Thank you, Vanessa. So I'm Judith. I'm located in Montreal, Canada. I'm part of Canada Bioimaging, VINA, and QUAREP. So because I'm based in Canada, I want to respectfully recognize and acknowledge the relationship that the First Nation, Inuit, and Métis people across Canada have with the land that all Canadians live on and enjoy. I recognize human rights of people who have suffered for systemic racism and discrimination in Canada. I seek to provide a welcoming, engaging, and safe community for everyone. And I'm also taking this opportunity to publicly announce that the Digital Research Alliance of Canada has renewed the support for the Canada Bioimaging Image Data Resource. That's our cloud-based Omero instance. And we're very happy to have their support for another good 3 years. So thank you very much. And now I pass the baton to Katrina.
Yeah, just briefly, I want to introduce Jens, who is going to give the talk today about the effort that they have been making to facilitate metadata annotation in Omero for the community. Jens is based at the University of Munster and is affiliated with the imaging network for microscopy imaging network of the University of Munster. He's a data steward, and his work is supported by NFDI for bioimage. So Jens, please, the floor is yours.
Okay, thanks for the introduction. Wait, I'm gonna share in a second. Correct screen. I'm simply going to assume everything is working fine. Wait, I have to make this smaller. Now we go. The workflow I'm going to present today is actually already a bit older. I've done this last year already. Just as a FYI, the uptake is starting slowly in our own university, so I hope maybe get the word out and maybe more people are interested and provide me feedback how I can improve this. Let's get started. I'm going to present a lower entry barrier, user-oriented image metadata annotation workflow for Omero with a lot of buzzwords. So let's get into it and look what it's all about. We will reiterate over the basics just to be sure everyone is on the same level. I will only skim over this, as I'm sure most of you are familiar with the talking points. Why do we need research data management, or RDM? Well, to properly handle the rapidly increasing amount of image data in all its beautiful variants. How do we do this? By implementing the FAIR principle with findable, accessible, interoperable, and reusable. And hopefully all of you are familiar with this acronym, but I'm pretty sure you are. And the final question, how do we implement this in practice? As of right now, the open source image data repository Omero is accepted by the community with overwhelming majority as their Lord and Savior for this question. So if you have never seen Omero in action, we will have a very short look at it, just to be on the same level. Omero is a web-based tool, meaning you can access it via your browser. And then you would look at something like this. In the left pane, you see your Explorer style tree view of your project's data sets, screens, and plates. In the middle pane, you see the thumbnails or previews of the images from the selected dataset. And in the right pane, you see all the nitty-gritty details, dates, descriptions, attachments, and most important, metadata. Now, this metadata implements the findability and reusability of the FAIR principle in the Omero implementation. In general, you can differentiate between technical metadata, for example, objective, laser line, exposure time, stuff like this, and experimental metadata. For example, what am I actually looking at? What is tagged? Is it a mitochondria, plasma membrane, what's the sample ID, the strain ID, what's the medium I use, the fixation, stuff like this. The technical metadata one can find under acquisition, a little bit hidden away. And the experimental metadata is supposed to be stored in the form of tags in key value pairs. Now, most of the experimental metadata in newer vendors, in newer vendor file formats, is already stored automatically by the microscope. Technical metadata, sorry. So Omero automatically via bioformats reads this out and stores it in acquisition. The user doesn't have to do anything. But of course, the microscope does not know what kind of dye you use, what kind of medium, etc. So the experimental metadata annotation load, let's say, is completely on the user side. Tags are just tags, or let's say hashtags, so single word or like very short phrases. And key value pairs are basically a combination, a pair of a key and a value. For example, here the key being the project and the value being the project ID. Now, it helps to have some kind of standard what makes sense to track for experimental metadata. So in 2021, some smart folks from the community sat together and proposed a guideline called RAMBI, a recommended metadata for biological images. By now, the majority of structured metadata workflows that I know of adhere to this guideline. And this is by no means strict law, but more of a community-driven process to distill a sensible baseline for metadata to start from for your own project. And it looks like this, summarized, which can be a bit overwhelming at first, but it is very helpful indeed. And most of the time, you only pick what makes sense for you and what you need. And the publication of RAMBI also includes nice examples. So you actually know what is meant by intrinsic variable, what is meant by experimental variables, experimental setters, etc., etc. So these were the basics. Let's get into the actual practical implementation. Envision the following scenario. A research group starts using Omero. And you, as a core facility member, give them an introduction for it. They immediately are enthusiastic and start uploading their newly acquired images onto Omero. And a week later, you decide to check on the Omero group, and all the images look like this. Zero metadata. Therefore, also not really findable and usable. And for the Omero admins and users in this round, if we are honest, 95% of our images look like this, no metadata. And therefore, the majority of the Omero content and image data basically does not utilize the fair functionality that Omero so generously provides. So it's kind of wasted. Now, why is that? We think the main hurdle for researchers trying to annotate metadata to their images is the time it takes to familiarize and execute with existing workflows. Let's take a quick look on the existing workflows or solutions for metadata annotation. First, of course, you can do it manually in the web browser, but even though you can do a bit of copy and paste, this is the most time-consuming solution and most painful, especially for large data sets or even high content screening stuff, which was what we had in mind when we designed this workflow. Then we have the native Omero Insight extension, MDE, metadata editor, the Omero Web plugin, Omero Forms, which has to be installed, and the Omero Web script key value from CSV, which will get a major upgrade from Tom, who's also listening, very soon, by the way. It's also natively installed. And then maybe custom implementations via Python or Java, but no, the most normal users will not touch those. And also all of them have their distinct advantages and disadvantages and are generally powerful tools. They all have one thing in common, their initial complexity. To visualize this, here we have the XML config file for custom MDE implementation. The JSON config file for the Omero forms looks pretty similar. And here's an example CSV for the key value from CSV script. And as you can see, it's not super intuitive or quick to create. It's by no means impossible, but you have to familiarize with them. You have to trial and error. You have to maybe have a little bit of knowledge about the semantics of JSON and XML, stuff like this. It's not ready to use, let's say, if no one else sets it up for you. So this is the issue we are trying to resolve with our workflow. Create a low entry barrier to achieve high user adoption and still provide as much functionality as possible. Broken down, the workflow consists of 2 simple steps. Annotate the image files with tags via the context menu in the Windows Explorer. And the second one, run an Omero web script. Enough about the theory. Let's have a look how this works in practice. And I've done it via videos because I don't trust a live showcase thing. Let's run. Here we have our folder with test images. We right click and tag the folder. In the little pop-up window, wait, I'm gonna actually pause it because it's going a little bit too quick. So you see the context window. There we have tag folders, which I'm gonna hit. And then in the little pop-up window, we just choose the tags we want out of the Rembi template we created. Basically, you run through it, or you can view it a little bit different, that you see the most used tags you most usually use. You can just search for tags as well. And if the tag doesn't exist, for example, GFP here, you can just create it on the fly and add it as well. And to finalize, we simply add the tags and choose here, tag the folder and all its files. And then we see a little progress bar here and the final success message. It pops up in the right corner. Okay, stop it. So that was the first step. Second step is the Omero web script. Here, for example, we see our imported images, test images with no metadata, no key value pairs, no tags. Then we select the overarching project. We select the script, annotation from tables. Everything is already filled out correctly for the initial annotation. And you just hit run script and watch it running. And to be fair, in the video, I just had to uncheck the tags box because I encountered a minor bug, but it's already fixed. So we see the script running for a bit. It'll be like 20-30 seconds or so. And then we get the final success message. Okay, annotated 80 images with 664 total key value pairs. Now let's have a look. And the image we had before is now properly annotated. There they are, the key value pairs. So this is how it looks in practice. How do we do it? Let's talk details. For the tagging in the Windows Explorer, we use a file tagging software called Tables. This is not exclusively for image data, it's just simply file tagging. You can tag anything you want to. Thomas especially uses it to tag every bit of information that comes in. It's really working great. Those tags can be shared and synchronized between different groups, for example, different research groups, different users, and different workstations as well. So you see the same tags in the microscope workstation as you see in your personal laptop. You can set up elaborate auto tagging rules. So you just put files in a folder and they get tagged automatically. You can have nested tags, which allows us to recreate the Rembi template. And here comes the most important part. The tagging data is stored in a Microsoft SQL database, which runs externally and therefore can be accessed from Omero. Why is that important? Because every other file tagging software we tried stores the data basically the tag data in the file name, which kind of sucks, or in a companion file, or a local database, MySQL or something like this. In any case, locally on the workstation, where we cannot access the tagging data from Omero. So we highly reliant on the external SQL database. Now we can create tags. The next question is how to use them. And we decided to also adhere to the REMBI guidelines and implemented them as a template in tables, which is fairly easy. We have 3 levels: the REMBI category, the key name, and the actual value. For example, for specimen would be the REMBI category, die would be the key, and DAPI, FITSI, GFP, for example, etc., are the possible values for the key. This structure also allows for, but does not force, the implementation of the Omero mapr plugin with the REMBI categories as different namespaces. I'm gonna talk about this in the next slide. Well, so much for the first part, the tagging. Next, what is the Omero web script doing exactly? Basically just queries the SQL database where tables stores the tagging data and resolves the tables tags with a little bit of logic into Omero map annotations, also known as key value pairs, and tag annotations, also known as tags. And we also, in our instance, we utilize the Omero mapr plugin to create those neat paragraphs that you see here of key values with the appropriate headers above it, which looks just nice. And also the plugin gives us the possibility to create thumbnails with hyperlinks. For example, here, this little thumbnail leads directly to the PubChem site for atomoxetine, I think it's pronounced. And Omero mapr also implements a search function for the values. Next, no, not next, but basically finally, how it all connects. The workflow. First off, you acquire your images on a microscope and have them on the workstation. Microscope workstation. Then you can tag with tables already on the microscope workstations, which saves the information in the SQL database. Next, you upload the images to Omero or to your local server or file share. And finally, in Omero, you run the annotations from table script to create the key value pairs and tags. Now, if you later on decide to add some more tags or change them because you made a mistake, you can do so from your own laptop or workstation in your lab or in your office, because the tables tags are synchronized between those machines, because all of the machines, all of the tables versions access the same Microsoft SQL database. Simply rerun the Omero script afterwards and the changes will take effect. We're also already at the second to last slide, which will take a bit longer because I'm going into a bit more details. And we're somehow familiar now with the details of the workflow, and let's look a little bit at the pros and cons. It's really just 2 intuitive quick steps, which was the main point of our implementation of this workflow. Make it easily accessible for users. It doesn't only support key value pairs, but also tags, which is nice. And if you're doing, for example, in-place import, the script could be run automatically after the import. The detailed auto-tagging rules can create the majority of tags automatically. Let me go a little bit into depth here. For example, you're always looking at Drosophila, you're always looking at a specific cell type, and you're always using DAPI to stain the nucleus. So you set up on the microscope workstation, you set up a folder with your name. I say, okay, within the folder, you do the auto-tagging rules. Everything in here that gets created in here or gets shoved into here, copied, pasted into here, already gets these 4 basically Drosophila tag for the organism. Maybe we always look at epithelial cells, then we have the organism part, the cell line might be the same, the strain. So you can cover 80% of the tags that are relevant under REMBI already with auto-tagging rules if you know your project is gonna revolve around the same cell type or the same setup anyways. So from day to day, you just tag the created images with the 2 or 3 missing things. For example, you added a new fluorophore, you fixated them differently, I don't know, you added a little bit of glucose, something like this. So you add just 2 or 3 tags every time, and the 80% of the tags are already there. Those tables tags, as I said before, are synchronized and shared between different workstations and groups. And as I also said before, you can update the key value pairs and tags at a later time. Now, disadvantages. Tables is a proprietary software, which is, I know, a red flag in this open source community. We really would have loved to find an open source solution which does the same, but we couldn't. But to be fair, for tables, the licensing fees are really modest. You're not going to go broke. So if you run a research group, you're going to need, I don't know, 2 or 3 licenses, and it's going to cost you, I don't know, $40, $50 a month or something like this. Or you only run 1 license, which is also doable under certain situations. So you run, I don't know, €7 a month or so, €8 a month. So it's not huge sums. Tables, unfortunately, only supports Windows, which is a downside for most personal workstations, as MacBooks are still a thing, it seems. But to be fair, most microscope workstations run Windows, so you could at least tag stuff already on a microscope workstation when you acquire the images. In the long run, what we hope for is that the workflow kind of gains interest and someone in the open source community replicates the functionality that Tables provides in an open source solution. But it's eine fromme Hoffnung in German. It's a hope. We are faithful. To also be fair, the initial setup of the SQL database, if you also use Omero MapR, are gonna take a little bit of time, but this is basically on the side of the IT guys or the sysadmin. And I would say I'm moderately competent, and it took me like 2 or 3 hours. There are very good step-by-step things out there, step-by-step tutorials how to do it, and I also try to explain it the best in the GitHub README I put up. So if you run into problems, just ask me, Google around a bit. It shouldn't be too hard. It's really well explained. There are some limits for tag naming conventions, namely you can't start with an underscore or something like this. So, but those are fringe things, and it is not providing all the in-depth functionality Omero Forms or Omero Inside MDE have. But this is a compromise. Our focus was the high user adaptability, high user adoption with like a low entry barrier. So I think we kept 80%, maybe 70% of the functionality of Forms and Inside or MDE. But of course, those things are more machine readable. They have a versioning stuff. And our hope is that this is like a foot in the door. People can use this, see, oh, it's easy and it's quick. But somehow it's missing some kind of functionality that I really want to, and then see, okay, what else is out there? Oh, it's Forms. Oh, maybe it's a little bit more complex. But now that I see the benefits of having metadata annotated images, now I'm willing to embrace a little bit higher complexity of Forms and MDE, for example, and set this up properly so I can have the higher functionality. And I think that already concludes it. Yeah, the disclaimer, I'm paid by the NFDI for bioimage, which is a project of the DFG, German Research Foundation. So this whole project is a contribution for NFDI for bioimage, and I'm a data steward there. So if you have any other questions, I'm also fairly active on ImageSC. Just hit me up. The code, the README, everything, the script is on GitHub. You see, them will maybe be forked or migrated to the NFDI for bioimage GitHub pretty soon. And the slides, the PowerPoint and the PDF of it are on Zenodo. with the DOI there and the QR code with there, or you just search for the title. I just put in the brackets there. Are there questions? Now I can actually end the slideshow. I can stop the share rather. So.
Thank you so much. This was very, very nice. Big virtual clapping. So there are some questions already in the chat, and anybody else that hasn't, I mean, anybody wants to, maybe we can just go through them so that everybody can have a chance to hear the answer. Otherwise, if there is any other questions, please feel free. So anybody wants to start?
Should I just read through the chat and answer the questions from there? Or?
So Glenn has raised their hand, so let's just from him.
Okay.
And then we can. I'll read the chat.
You can do the chat first if you prefer. I don't
No,
mind.
no, go ahead, go ahead.
Well, it was sort of two questions. I like the solution. It looks nice because so certainly using MDE, I don't find that particularly easy, and I sort of know what I'm doing. So my users have no chance. I was curious as to, firstly, if you've got nested TIFF images, such as Leica image files, does the tagging work, and does it tag each individual image inside that LIF when it gets imported into Omero? Because that then splits them up and gives you each individual one. And secondly, do the users need to have write access to those data when they're adding the tags, or can they do that without having write access to that folder?
I'm pretty sure if you can see it and can right click it, because you're not changing the actual file, you're just creating an entry in the SQL database.
Okay.
I didn't try it, to be honest, but I can maybe answer the LIF file thingy. Can I share the screen just again?
I guess with the LIF files this will not work, because tablets is made for files on the computer. So the LIF file is one single file, and you will text this single file. Yes, and then you will not have it.
Yeah, that's what I was thinking.
So basically, we have a LIF file here for everyone who's fairly familiar with Omero. So I click it, the file set kind of gives it a light blue tint. So all of this is one file. If you go into the path and say imported from, that's like a one LIF. And what the script does in the background, it basically gets the path, the imported from, and then just checks the SQL server, okay, is there any entry for this path? And if yes, give me back all the tags. So with this, basically all the LIF files in here have the same tags and key value pairs because they all have the same imported from path. I hope this answers the question.
Yeah, it does. And if you have them tagged, if they tagged them, for example, on the acquisition PC before the data got moved to the network drive, do the tags follow it or not?
You can set up a rule where you kind of synchronize different folders to have the same tags. But for now, this is one of the drawbacks where it relies on imported from path. For now, I have not yet found a better solution for this, but it's high up on the to-do list to find a better solution if people move things. Yeah, I'm fully aware of that.
Mm-hmm.
But you can set up, if you always move it in the same folders, the same structure. If you have the same structure in the microscope workstation and on your file share, you can set up a synchronization rule in tables that basically kind of cuts the path before and just pretends everything. Oh, I'm missing the right vocabulary. But you can.
I know what you mean. It removes the first part of the directory.
Kind of, yeah. You also can do this, for example, if you mounted a file share different on different workstations. If it's somewhere it's a set file share and some it's a Y, but the other path is completely the same. You just set up the synchronization, say okay, set equals Y, like very naively speaking, and it says okay, cool. then it works.
So Thomas wants to maybe add something?
Yes, so as long as you haven't imported the data, so we are using Omero Insight still at the moment. So if it's, so tables is running on the acquisition computer, and if you then move your data, it's watching. So if you move the data from the acquisition PC to a network share, tablets will recognize this. But once you have uploaded the data with Omero Insight, then you have this path. Then you shouldn't move your data anymore. And then you need something like the synchronize, what Jens explained. But as long as you have this auto tagging directly at the acquisition computer, and then move it to your network share, that works. And then uploading it, that works.
That sounds good. The reason I was asking about write access is the network drive we have is deliberately not writable by the users. So they can't delete the data without going through Omero. So yeah, thank you.
So before reading the questions on the chat, I just want to clarify. So I think now I just wanted to clarify that I got it correctly. So pretty much images are acquired and they are now on the acquisition computer. They are there, the user will tag them, and then it will do the import, and then it will run the script. So they have to be tagged before import.
You can tag them after import as well.
But not
as long.
Oh, wait.
Yeah, I mean, I think the most likely scenarios, people will tag them on a microscope because they're sitting there anyway for 2-3 hours, and then it's like, for the time being, I can already set up the tags and everything. But if you don't want to, or it's not connected to the network, for example, your microscope workstation, you can also do the tags later on your personal workstation, your laptop, or in the lab.
And then you run the script.
And then run the script, yeah. But you have, of course, you have to tag it first before you run the script.
Yeah.
And then when it is imported in Omero and the script runs, it for now the hard limit is that is really looking for this imported from path. So before, as Thomas correctly said, kind of tables follows it around and changes the path internally in the SQL database. But as soon as you import it from some path, you better leave it there. And you better change the tags on this path.
Yeah. Okay. So one question that was raised was, but I think you already answered, is the fact that it's not available for Omero. For Mac. But it is available for Linux. No.
I think not. No.
Okay, so it's only available for PC. Okay.
Yeah, they purely did it on Windows, unfortunately.
So then there was some discussion that Damir had. Maybe Damir, do you want to talk about what you were saying? Because it's being run primarily. So the point is that this might not be such a big problem because most acquisition software actually depend on a PC. So the acquisition microscope typically is a PC. Anyway, so that was the discussion that was there. So the other question was basically the import. In this case, the tagging and the import are two separate processes. One tags and then imports, and then once you're done, then you run the script and the two things are synchronized. So that was another question that was asked. So then Moshen asked about, whether there is an automated script for import, and that was already answered. Okay, and then there was a question about the connection with ELN. So maybe this could be, what is the integration with ELN, like LabArchive or SciNote? Is there any integration so far? If you want to answer that.
We have not thought about this. To be fair, I don't know LabArchive or SciNotes. We just set up eLab for the World in our university, so I'm starting to play around with that. I think if there are open APIs that you can access with Python, you might be able to play around with stuff, because the Omero web scripts rely on Python. And yeah, I'm just spitballing here. You might be able to do stuff, but I'm not sure.
But I mean, there is an API, so you're using the same. Yes, we are just going to the database and grabbing the information. And this you could also do on a script you're running in ELN. So this should be the same. Then you just need a path or something to the file in your ELN. So if you have the path, then this would also work. You need just this information. But,
yeah, the issue with the ELN,
we haven't done it yet. So
I wonder if Alexander wants to ask his question more specifically, what he was thinking of. But normally ELN that I know of, they're not necessarily aware of where the images are. So that would be the issue, is because go ahead. Who wants to
yeah, I mean, many ELNs like offer REST APIs to, exchange data. And I mean, if you see any, or maybe if tablet, has some kind of REST API that could be used to send all this metadata to the images, maybe like at least to the experiment. Maybe you just run an experiment and you can like want to duplicate some of this, metadata into your nodes on the experiment.
Tables themselves don't provide an API. I think they had I think they had a prototype, a beta version of some kind of API, but I would have to check on their website. Basically, you would have to fiddle around with the SQL itself, with the database itself.
And another question, like, do we need to pay any license for Microsoft SQL database server?
No. It's a good question. Good point. I actually forgot to mention this. It's also proprietary, of course. It's Microsoft, hey. But the free version is completely enough to service whatever needs from tables may arise.
And what kind of server it would need? Like,
I don't know what the free tier is called. Oh, you mean a virtual machine? Oh, it's I don't know. We have it on we have it on a Linux virtual machine. It's not a big thing. A really small thing, like four CPUs or something like that. The SQL database, it doesn't really. It's not resource hungry.
But it's something that should be running all the time in the background.
Should be, yeah. We have it as a virtual machine in our university cloud. So as soon as you're in the cloud, you can access it. That's also the whole point, that each research group sits in another building, but needs to access this external database. And Omero, of course, the virtual machine for Omero sits in the server room, but has to be able to also access this external database.
Okay.
And all the communications encrypted. Do you know how well they encrypted?
You do it, the communication from Omero, or when Omero queries it, it's ODBC driver, so I'm not quite sure how encrypted it is. I would have to go into the details. If you're more familiar with this, maybe just check the source code and you can find it for yourself. I did not have a specific focus on encryption there, to be honest. It might be just as a default, but I didn't like purposely put it there because it's all in a university network and it's some SQL database entries, file paths and tags. It's not really.
Maybe
dangerous.
we can firewall it or whatever.
So then there was a just to continue on the chat, if this Josh proposed that there might be something, maybe Josh, you wanna say what you mentioned?
This isn't too well formed, but just anyone else who's interested in doing something similar open source, that might be an interesting conversation for what it's worth. In one of my projects, which uses Globus for uploading everything, basically what ends up happening is the users start throwing things in directories and they each have their own hierarchical storage. And what I really, really want is the ability at each of those hierarchy levels to gather some rule from them. So essentially what Jens and Thomas are showing and say, you know, at this level, tag all images that get created under this directory with this experimental factor. But everyone's going to have it slightly differently. So I don't want them to have to create a spreadsheet or configuration file, just some super simple way at each level of their hierarchy to add a tiny little bit of metadata. And that would actually go quite a far way in being able to automate what I want to do in Omero. But I don't know of that system, and I've always been just short of actually starting to produce it. But maybe if there are enough of us who are interested in exactly this workflow that Jens is showing, then it makes sense to go ahead and do it. Because I think that happens pretty frequently.
So the idea is to create something instead of tables that is open source.
Yeah, and I think so. That's the other part of my statement is I think there's a benefit to doing it in directory space and not having that extra burden of the database. There are some downsides of doing that, but it's just kind of a fundamentally different model. But I think Omero could be aware of those things, and you know, as you kind of go down the directory hierarchy, you could pick up all the little bits of metadata and produce something at the bottom, right?
Mm-hmm. Okay. Thomas wants to say something?
Yes, so we are also totally in favor to have an open source and maybe a little bit more for our purpose streamlined solution. So we tested it. We did it also now in a course with this auto tagging that we had, for example, on a Zeiss Elira 7. So this is one folder. So all tags, every image which is produced on this gets already the tag with a microscope. Then we have some folders for the function like turf mode, aperture mode, zim mode, whatever. And you're tagging this directly automatically. The user doesn't have to do something. This works really great. And to have something which is just tagging the files, that in theory, it's not based on Omero, so you don't need Omero for it. You can also have ELNs or whatever to get this metadata, because students don't like it to do it again and again. They have their ELN, they have Omero, they have something else, and they don't like to add the metadata in all of these parts because it's duplicating work. And that was the idea behind it. So tables is not perfect. For us now it's working really good, but we have to see for other solutions it might not work. So totally in favor to have something which is open source and for everything, like Linux, Mac, also available. Yeah.
So, so you were saying, actually this reminds me on a question that I had, which is, so you have launched this a while ago, and so what can you tell us a little bit more about how your users are interacting with it? They like it. You started seeing more annotation popping up. Can you tell us about that aspect?
I can, but it's gonna be short. We introduced it to one group because they had really issues. First, they were, as I said, enthusiastic, and by now, but I don't know. We gave them a couple of licenses. They tagged stuff, but I have not heard a lot of since then. Normally, we have a good connection to them. So if something would have gone wrong, or issues or bugs or something, like, they would have come and said something. So I think it's just it's a slow start. It's a slow uptake. Wherever I go, I try to promote it and show people, okay, this is really easy. Do it, please. But yeah,
I mean,
it's a slow start.
I guess it's sorry, I guess it's the same story like with Omero. So it comes from bottom up. So you always need some people who starting with it, and then it takes quite a time until all of the groups are doing it. So we have, so in the student course, it was working quite well. They liked it, but if the group is not using it or the PI doesn't say you have to do it, then it will be very slow to coming up. So yeah. But this is the same experience with Omero in our.
Yeah, one thing that that I would say, I mean, I haven't tried it or anything, is just how can, because what I can understand now is that every time the user has to re-tag everything. So basically is there any way, like you were talking about auto tagging, which is like, for example, you know, the fact that if you're coming from this specific workstation, which is connected with this specific microscope, let's at least that tag is there all the time. So, but the way it is now, unless I'm mistaken, every time there is an acquisition, they have to retag. Is that correct? I mean, every time there is a new acquisition, they have to assign tags again to the images.
If they did not set up auto tagging stuff, yes. If they know,
so how does it work, the auto tagging? So that I didn't understand. Like
maybe I can show you. Wait.
Yeah.
Did I do screen one? Zack. Let's make
so
a new
if they
folder.
keep the same structure, then it will be auto tagged.
No, I think it's just Windows 11 just kills it. Just a hit, image data. I'm just doing stuff. Where was it? I think it was in the last thing. Create auto tagging rules. Files and subfolders. That's it. Zack. Now I just copy paste stuff. Oh no, it wasn't size, it was the new folder. And it should have the tags. Okay.
But so basically,
that's why I'm not doing live presentation.
It never happens when you're trying to do it, you know, like, yeah. But no, the point that, so just to understand, so if the auto tagging is based on the fact that if you have the same name of directories, they will always receive the same tag. Is that how it works?
So
like,
if there's a file created,
as mutant and images in mutant and wild type, for example. Two directories, mutant, there's certain date, and then inside mutant, wild type, and then inside images.
Yeah.
Will the mutant and wild type always receive the tag, the correct tag, because it's always using that?
Whenever a file is, if the auto tag, or if the rule is set up, whenever a file is created in the mutant folder, it will get the tag mutant.
Okay.
As simple as that.
So basically, there are a lot of rules you can assign. So you can assign one folder, assign some tags, and say everything which is saved in this folder or subfolders get these kind of tags. Yes.
Oh.
And you can also do it on the file name. So if there's a file name or a file which has a name with mutant inside of the name, it will get the tag mutant. All CZI files, so with an extension, get size microscope.
Yeah.
So there are also regular expressions you can use. So a lot of rules you can set up. However, you have to set them up beforehand. Yeah. So this is some work. But
okay, that's great, because then if the same group always uses, for a certain series of experiments, uses the same tagging, not the same directory structure, then they don't have to go through and re-tag everything. Okay, yeah, that makes sense. Okay, any other questions? So somebody came in later, so maybe Jens, if you can put some links to your.
Of course, yeah.
So that they can, and Natalie, go ahead. So that
just curiosity question. So You didn't have much success with your user using it so much, but have you had, do you have any example of people wanting to find this data and reuse it for other purposes using the system? And how would they go about it if they wanted to do that?
I think this is a higher level question. If they want to find and reuse it, basically what you have to do is make it I'm gonna close my window. Make your Omero instance public. Make it as put it in a public group.
But let's say it's you said like everyone at your university, all the different groups have access to it.
To the tables? Yeah, they can use it.
So let's say they want to.
But yeah. But also you have to you have to separate the groups a little bit. For example, one group always runs experiments on Drosophila, the other one is on yeast. So they have also different ontologies and different tags they have to use. So if you always see all of the tags of all possible different research groups, it's going to be just a clusterfuck. So what you do is basically you do for each group, you give them the Rambi template, you kind of populate them. So it's an easy, once it is done, the Rambi template, you populate it with examples, DAPI, stuff like that. Basically almost everyone uses, and then they put in their specific keys and values that they use. So they're not overwhelmed with the possible tags and keys. And might also be that tags might mean different things for different model organisms or different contexts. I have no specific example for that, but I can imagine there's a fringe thing where this happens. But if you want to make it like openly findable and usable, you have to put it in an open Omero group. But it's easier if you already have all your image data kind of in an almost automatic way metadata annotated, and then just shove it as like, that's my publication. I use these 500 images in these 20 data sets, and then just say duplicate these 20 data sets, which are already like at 80% annotated how I want them to be to publish them, duplicate them into the public Omero group, and then it's done. So you don't have to, oh, now we want to publish, these are my 200 images. What were those images again? And then you have to go through each one of them and like kind of manually annotate them. I see hands raised. Is this answering your question somehow, or did I miss the point?
Sort of, it's getting there. It's a hard one.
Okay.
So, Thomas, and go ahead.
Okay, let me try. So basically that goes away from tables, but that's fine. So we have it with older PhD students. If the PI is looking for a paper and the PhD student already left, so at the moment also with not so much annotations he could find it. So there Omero is already a big advantage, but you can then search for key value pairs for the text and everything like this. Basically, it's a game with the tags and key value pairs. You don't have to know where you saved something. You just have to know what you're looking for. So what is it about? Because you have a description of your file. It was a mutant, it was a cell, we stained it with Alexa, and then you can click all of these things, and it doesn't matter where it is saved, you will get all of the files with this. This is the idea behind this metadata. For reusable, they're basically, so we haven't had this a lot yet, but so where we have data, which we would like to publish, exactly for this point is screening data with yeast for deep learning. So basically this is coming more and more. And if you think about just let dream, all of our data is tagged perfectly and all of our Omero instances can speak with each other, or everything is saved in BR or IDR, doesn't matter. And then you have a lot of training data for everything. Then you're just uploading an image and the data comes automatically, yes? So it's this is but just one really good example. So because the bioinformatic guys are not producing the images, they cannot redo the experiment, and then they need good metadata to know what it is about.
Yeah, so
that's where I was going. So thank you, Thomas.
yeah, I think the key is how to get people to annotate their data, thinking about the future. So the way that I am and other people are, thinking about it is to sell it to the people that are producing the data, because they have to do the heavy lifting of the annotation. Is a gift to yourself first. I mean, before even you think of other people, can you find your images when you need to publish? Everybody knows that experiments may last, projects might last more than 1 year, 2 years, 3 years. When you have to put everything together, can you find it? And that is a pain point that everybody has experienced. So forget about even, now the NIH is asking for, sharing and all of that. So that's another, but a lot of people are still in denial on that, but at least everybody knows that they have to publish. And just even for publishing, making a gift to yourself so that you can get these images later on, and then actually be efficient in producing the figures and stuff. And so, for example, with some of our users, that is the main selling point, and they really like that. So it's more like for organizing their own data. And then you also add the value, by the way, if you do this, not only can you find them, but then you'll be in a much better situation when you have to share. Anyway, but the key is, so that the people that what everybody here is trying to work on and find a better solution, best solution together. That's why we created this user group, is how do you make it as easy as possible for the people that are the producers of the data. Anyway, so this was super interesting. Thank you so much. I want to tell you that we have our next our next presentation is going to be in June. A lot of us are going to be at AIMM, but the presentation is going to be given by Alex Regano in my group, talking about the project is working on to do annotation upon import. So doing the annotation and the automated import at the same time. So I'll tell you about that. And this is just the idea is to share all and find a better solution altogether. So I can't remember right now what date it is. Maybe Vanessa can tell us if it's going to be the Friday of the first week of June. Okay, so I'll see you all next time. Thank you so much. Vanessa, do you remember the date?
June 7.
Okay.
Yeah.
Great.
Tuesday,
Thank you so
first
much.
Tuesday in June.
Thank you for your presentation. This was super interesting.